Okay, now in this video we're going to see how we're going to process this nitrogen by the liver so that we can get rid of that nitrogen before it builds up into any form of toxicity for the body. So the way the liver is going to be disposing of nitrogen is through the urea cycle. So the urea cycle is exactly that, it's a cycle, so it's going to come back on itself. So we have to do a series of reactions in order to take the nitrogen coming out of these transamination and deamination reactions and capture them into chemical structure that we can process. And the end goal of this is to synthesize urea, which is then put into the blood and filtered out by the kidneys. Okay, so now recall about our free nitrogen that we talked about in the mitochondria in the last video. So those two-step reactions of glutamine being processed by glutaminase to release free nitrogen as ammonia, and the second step of glutamate dehydrogenase that is going to be releasing a second molecule of ammonia. Those reactions are occurring in the mitochondria. So part of the urea cycle is in the mitochondria, and part is in the cytoplasm. Now there's a distinct reason for this, because there are two entry points for nitrogen into the urea cycle. In the mitochondria, the entry point of nitrogen to form urea comes from those reactions that are releasing free ammonia. In the mitochondria, in the cytoplasm, the other form of nitrogen is coming in as aspartic acid, aspartate. This is typically formed in the cytoplasm, and it is going to contribute a nitrogen to the urea cycle. These two nitrogens themselves will form urea. So one nitrogen of urea will come from the free ammonia. The other nitrogen of urea will come from aspartic acid. So now, this being said, let's look at the construction. Let's see how the free ammonia is captured and also aspartic acid is processed. Now, aspartic acid is easy. Recall the buildup of glutamate, because remember I said glutamate is the reservoir for amine groups that are being transferred off of amino acids to make their respective alpha keto acids and transferred to glutamate. Remember I said we had to restore alpha keto glutarate in order for these reactions to continue. Well, the way this is happening is that we are taking the amine from glutamate and we are putting those amines onto oxaloacetate. This is a transamination reaction. So oxaloacetate is the alpha keto acid of aspartic acid, and alpha ketoglutarate is the alpha keto acid of glutamate. So a transamination reaction is happening by the enzyme AST, which is aspartic acid transaminase. Now, this reaction is happening in the mitochondria. Remember, because we have a lot of glutamate in the mitochondria, so this is another reaction. The aspartic acid then comes out of the mitochondria and into the cytoplasm to contribute that second amine into forming urea. Okay, now let's turn our attention to the glutaminase and the glutamate dehydrogenase reactions in the mitochondria. They are generating, if you hear noise in the background, that is my large male cat having a wild cat moment. And he's tearing up a whole bunch of papers in the corner. Okay, back to the topic. We have ammonia being generated from these two reactions, and we need to capture these as quickly as possible because ammonia is toxic to cells, and it's very toxic to neurons. Ammonia can leak out of the liver, get into the blood, and it's very soluble in the blood, and it gets to the blood-brain barrier. It crosses the blood-brain barrier with ease, and it gets into the brain and can cause coma if it's too high of a concentration. So recall in the last video, I said that as soon as the nitrogen, the ammonia, is formed by these two reactions, it is captured essentially immediately. And the way it's captured is through bicarb plus ammonia and we are going to form from this a molecule that is called carbamoyl phosphate. Now this molecule here, let's take a look at this reaction. Obviously you can see a phosphate here. This is a high energy phosphate bond, and so ATP is going to be hydrolyzed, and that energy is going to be captured in the carbamoyl phosphate structure. The enzyme that does this reaction is called carbamoyl phosphate synthetase. So now notice the bicarb. Where does the bicarb come from? Well, recall what is generated during the oxidative energy production. TCA cycle, we're generating carbon dioxide. The carbon dioxide is converted to bicarb by carbonic anhydrase, and then this structure here is the byproduct of respiration, and normally it enters the bloodstream and red blood cells and is transported to the lungs in order to expel the carbon dioxide, but in this case the liver is able to use it in forming the carbamoyl phosphate structure. So this is a very, very rapid reaction, and again, as soon as the ammonia is formed, it is captured by the bicarb because there's sufficient amount of respiration going on to form carbamoyl phosphate. Once it's in the carbamoyl phosphate form, the nitrogen is now essentially neutralized as far as any potential harm. Now the next structure, the next reaction, is we are going to take our carbamoyl phosphate and we are going to react this with an unusual amino acid. It's an amino acid that is not found in proteins, but it's an amino acid nonetheless. And this amino acid is called ornithine. So notice it has an amino acid structure. It's got an alpha carbon. It has the amine, primary amine on the alpha carbon, carboxylic acid, and it also has an R chain. So this is ornithine. Now we are going to combine these two reactants, and we are going to form a molecule of citrulline. So here's our amino acid backbone. Here's our backbone structure of citrulline. Now we come down here to the end where the nitrogen is of ornithine, and we are going to add the carbamoyl phosphate onto here, onto that terminal nitrogen. This structure is called ornithine, citrulline. The enzyme that performs this reaction is called ornithine transcarbamylase. Now note that we've added our carbamoyl phosphate group onto the terminal nitrogen of ornithine to form citrulline, and look at this structure. Does that look familiar? Down here. You can start beginning to see the construction of urea here. Now at this point, this has all been happening, these reactions here, these first couple of reactions have been happening in the mitochondria. The citrulline is transported at this point into the cytoplasm. It's a an antiporter, and what is transported back into the mitochondria is ornithine. So this is an antiporter, and it's considered a 1-to-1 ratio antiporter, so that one citrulline is transported out of the mitochondria into the cytoplasm, and one ornithine is transported from the cytoplasm into the mitochondria. So from this here, you can already start predicting that the citrulline reactions that we will undergo in the cytoplasm will eventually come back around to ornithine. The ornithine will then enter the mitochondria and replenish the reaction with carbamoyl phosphate to keep the cycle going. Now, once we have our citrulline structure, we are going to add to the citrulline structure an aspartic acid. And this is the second nitrogen of our aspartic acid. So this nitrogen here, recall, came from the reaction to form carbamoyl phosphate, capturing that free ammonia that's formed. What we are going to do now is we are going to add aspartic acid to this carbon on citrulline. And what we will form is a molecule called arginino succinate. Here is argininosuccinate. It's a very large structure, but now what we've done is we've added our next nitrogen. I should put hydrogen here, and then this nitrogen here comes from the introduction of aspartic acid. And remember, the aspartic acid came from transamination reaction with glutamate. And then recall this nitrogen here came from carbamoyl phosphate. These two nitrogens here will be the backbone structure of urea. Now the next reaction is that we have added our second nitrogen to form urea using aspartic acid, and now we have to remove the carrier so that we can continue this reaction going, because we will continually need to unload the glutamate into aspartic acid to keep the urea cycle going. So the backbone structure that we are going to be releasing in the next reaction is this, because we've already donated the nitrogen that we needed. And this structure here is fumarate. Fumarate. So now this fumarate will be recycled, and we'll see this in a minute, of how this is recycled into forming additional aspartate. But when we remove this structure here, the enzyme is called argininosuccinate lyase. To remove the fumarate from the argininosuccinate. And now what we are left with is our amino acid arginine. So this is the amino acid that we all know, called arginine. Now the next reaction is simply a hydration reaction and in this hydration reaction is accomplished by the enzyme arginase, and we are going to liberate urea at this step. The water will add to this carbon and it will release our urea, and notice this structure here. When this is released as urea, what we have left is ornithine. The ornithine at this point will re-enter the mitochondria by the ornithine citrulline transporter. And by doing this, now we've restored the ornithine back into the mitochondria in order to continue the urea cycle by picking up more carbamoyl phosphate. Okay, so the ornithine is restored for our cycle so that the continued scavenging of free ammonia is good. But what about the aspartic acid, which is contributing the second nitrogen to the urea cycle? Well, recall fumarate is generated from the cleavage of argininosuccinate by the argininosuccinate lyase in order to form the amino acid arginine, which goes on to form urea plus ornithine. The fumarate now is able to convert into malate in the TCA cycle. So at this point, the fumarate converts into malate by fumarase. The malate converts by malate dehydrogenase into oxaloacetate by the redox reaction. The oxaloacetate now, remember, is the alpha keto acid of aspartate. So this transamination now can form aspartate by the AST transamination enzyme, and now we've regenerated our second nitrogen donor to the urea cycle. This can now add on to citrulline, forming argininosuccinate, and continue the cycle going. Okay, so that's the basic reaction sequence of the urea cycle. And now the next video I'll prepare is just strictly amino acid metabolism, and I'm going to show you an abbreviated way of thinking of this, because amino acid metabolism is very complicated with all 20 amino acids under consideration. So we're going to talk about how amino acids are broken down, how they are formed generally, just to get the general concept across.